share my screen.
There we go. Okay. There you go. You're up.
All right, good. Well, thank you. Thanks, Melissa, for introducing this. We'll spend the next 45 minutes or so talking about some of the things we're working on, and then I think should be time for questions at the end. What I want to start with today, though, let's see if I can get this to actually advance. All right, there we go. I just want to take a minute to recognize the passing of Anita Oberhoster. Many of you will have seen this story in Wine Business Monthly or other places. At last week, she passed away about 10 days ago. And there have been a number of stories, but I just want to really focus on how key she has been to smoke taint research in the United States. We started working on smoke taint here in 2015, 2016, really the first project. She's been an active researcher in the area of smoke effects on grape and wine quality since 2017, since the first really big fires in Northern California. She's one of the co-PIs, along with me and Elizabeth Tomasino, on the SCRI smoke taint grant we had starting in 2022. We're just beginning the fourth year of that grant now. As Melissa indicated, she was one of the university researchers on the West Coast Smoke Exposure Task Force since the inception. And I think it's actually been more like 7 or 8 years, because it was pre-COVID when we started this. And of course, she's been part of the projects funded by USDA, ARS, congressionally allocated funding over the last 4 or 5 years. I think we're in year 5 of that funding. So just a huge player in this area, a terrific colleague, longtime friend. She's definitely going to be missed. So I just wanted to spend a little bit of time recognizing her efforts in this area. All right. So what I want to do today is just talk about some of the things we're doing. I have a list here of the research areas that we've been working in over the last 2 or 3 years, things like barrier sprays, mitigation in the winery using nanofiltration and enzymes. You can see the list here. What we're going to focus on today are the first 4. We'll spend a fair amount of time on barrier sprays and mitigation in the winery, and then talk a little bit about our sensor networks and some of the work we're doing with RNA transcriptomics and metabolomics. This project was actually funded initially with seed money from the Wine Commission. So although it's largely funded now by USDA, I think it's worth going back to and seeing where that seed money has led us over the past several years. So I'll start with barrier sprays. We've been working in this area since 2018. But what we're talking about are materials that can be applied before a smoke exposure, maybe in some cases in the early stages of a smoke exposure, with the goal of making some kind of a barrier, or in some fashion preventing the compounds that are present in the smoke from reaching the grapevine and more importantly, the clusters, the fruit. We've worked over the past several years with kaolin and bentonite. We make slurries of those 2 clay compounds and spray those on the vines, with the goal being that they're both strong adsorbents, and that perhaps they can remove some of the smoke compounds before they reach the fruit, take it out of the smoke plume, adsorb it into the materials, and prevent it from getting to the fruit. So we've been working that area. There are other groups working on polymer coatings, things that would provide a physical barrier, things that would stretch as the berry grows. These may involve absorption as well, depending on the type of polymer. Elizabeth Tomasino and some colleagues at Oregon State are working in that area. Anita was actually doing some work with barrier sprays as well. But what I'll focus on is our work using kaolin and bentonite. Kaolin was first tried actually in Australia. Kerry Wilkinson's group looked at it and published a paper showing somewhat mixed results. It could reduce the concentration of volatile phenols if you used a kaolin slurry and sprayed it on the vines. How effective it was depended in part on the application rate, so how much you applied and how well you got good coverage on the fruit and on the vine. She also reported a bit of a varietal effect, that it seemed to work better in Chardonnay and Merlot than it did in Sauvignon Blanc. One of the key things from that study from 2019, the Hulst et al. Study, was just how quickly, after a smoke exposure, the compounds get converted to glycosides. They showed within a day of the end of the exposure, very little free glycol or other marker compounds were present. It had all been converted to glycosides in the exposed fruit. One of the key things that wasn't clear from the paper was whether the kaolin was removed before they did any analysis or winemaking with their fruit. And you'll see why that's important in a minute. So we started working with kaolin and with bentonite in 2018. I had support from Murdoch Charitable Trust that provided some summer help with a local high school science teacher, and we gave them a little project to apply these materials to vines, look at the fruit to see what was there, which he did. The 2018 study was really inconclusive. There really wasn't an effect between control and sprayed fruit. We repeated it in 2019 to finish his grant. But the difference is in 2019, in 2018, he made a whole berry extract, just homogenized everything and looked for the compounds. In 2019, I had him peel the skins off of frozen berries and just analyze the skins. Well, in the process of peeling the berries, he also wiped all the clay, all the kaolin or bentonite off of the skins before he did the extraction. And when you do that, you get a different result. So on the left-hand side are our control vines, CX, CK, and CB are control vines, either unsprayed or sprayed with kaolin or bentonite. On the right are our smoke-affected fruit. These are from the smoke houses that we use in our trials. SX is the unsprayed control. So that's what you get if you don't do anything during this smoke exposure. And then you can see with both kaolin and bentonite, SK and SB, we see a reduction in the amount of glycol of 30 to 40% compared to the unsprayed control. So we were seeing success with it in this point. 30 to 40% may be enough to take you from fruit that's smoke affected to something you could actually use. So we continued it. 2020, with all of the smoke that we had in the vineyard, was kind of a tough year, but so we didn't really find anything there. But in 2021, we tried it again. And again, we applied kaolin or bentonite as a slurry the day prior to one of our scheduled exposures. We went in 7 days post-exposure. So we had some unsprayed control vines. We had vines that were sprayed with kaolin, vines that were sprayed with bentonite. We went in 7 days later, and on half of the vines, we washed as much of the kaolin or bentonite off as we could, knowing we're using a backpack sprayer if there's a limit to how much we can actually remove. So there's a little bit of kaolin left, but we got rid of a lot of it. Same with the bentonite. And then we came back at normal maturity, harvested the fruit, and made bucket scale wines, micro scale ferments with the fruit, and then did an analysis of free and total volatile phenols using GCMS. So that's what I'll show here. On the left is the free glycol, and we saw not the same numbers, but similar outcomes with 4-methylglycol in the cresols. And on the left, you can see in blue is always the fruit that was either sprayed or not with kaolin, bentonite, etc. But U means in this case unrinsed. We didn't wash the kaolin off. We didn't wash the bentonite off. We didn't spray the vines down. And then in yellow are the vines where we wash the kaolin off, wash the bentonite. And for the controls, we just wash that off with water. And you can see on the free glycol side, there's not big differences between any of the treatments. You see a little bit of a reduction where you rinse, but it's not huge. When we look at total glycol by acid hydrolysis, you can see in this particular on the vines that were the control, where we didn't put any spray materials on them and then either didn't rinse it or rinsed it, we're at about 170, 180 micrograms per liter of glycol. No difference really between the control that was unrinsed and the control that was rinsed. When we look at the vines where we had applied kaolin, there is a big difference between the unrinsed vines and the ones where we went in and washed off. So washing the kaolin off, washing the bentonite off, reduced the amount of glycol. And we think it's a simple matter of the kaolin adsorbed these compounds from the smoke, and if you rinse it off before you make wine with it, you're removing it from the system. The one thing that's interesting here is if you look at the blue, the kaolin unrinsed and the bentonite unrinsed, they both have higher numbers than the vines where we didn't do anything. And I think this shows we are adsorbing these compounds from the smoke. And if you don't take the kaolin off afterwards, it's actually then just releasing the kaolin that it's absorbed, or the smoke compounds it's absorbed, into the fruit. And so we end up with higher numbers than if we hadn't done anything at all. If you wash it off, we do see a reduction. And I think if we had a more thorough way to remove the kaolin or remove the bentonite, if we were using better equipment than backpack sprayers, we probably would see further reduction even from what we saw. So that the issue here is, if you're going to use kaolin, if you're going to use bentonite, you really need to get in after the fact and rinse it off. If you don't, you run the risk of actually making the problem worse than if you had done nothing at all. So that's our key finding. And I think this is consistent with some of what Carrie Wilkinson had reported in her study, that sometimes it worked and sometimes it didn't. I think if you're not careful about when you remove it, or if you remove it at all, you may find that it not only doesn't help, it actually can make things worse. So that was 2021. In our trial this past year, we picked it up again. One of the things we're looking at this time, we just focused on kaolin. We're not looking at bentonite in this study, but what we wanted to understand was, do we need to apply kaolin to the entire canopy to get an effect? Or can we just apply it to the fruiting zone? So if we just spray the clusters versus spraying the whole canopy, what do you get? And we did the same thing here. We went in actually just a couple days after the smoke event was completed and washed the kaolin off of half of the vines, again using backpack sprayers. Half of them we left the kaolin on. We have not yet finished doing analysis on these wines. We had, between a couple students graduating and one of our students being out for part of the fall, we just didn't have the opportunity to get through all the analysis. So it's ongoing now. So as soon as those results are available, then we actually have 4 years of this study and we'll be able to put a paper together. But we think that one of the key lessons here is, if you're going to use kaolin, if you're going to use bentonite, you have to go back in and wash it off after the smoke is cleared, which may mean you have to reapply it if there's risk of a second exposure, but I don't think you can leave it on the vine for very long once the smoke is cleared. So that's barrier sprays. I want to talk a little bit about the sensor networks that we've been building. So in this slide, you can see on the left is the big smoke plume from the events in 2020. So this is smoke from fires in Northern California and Oregon that actually went out over the Pacific Ocean and then came back into central and eastern Washington for a period of time in September. And on the right, you see the air quality sensor network that the state of Washington runs. And purples and reds mean there's a lot of smoke in the air. So purple in particular, those are AQI numbers 200, 250 and higher. Some of these, in fact, were off the charts from anything that we'd expected. So these are the networks, these are the sensors that are in place through the state of Washington, the air quality folks who run these sensors. And that's fine. It gives us a good idea of what's happening, but one of the issues we felt with these sensors is they're not necessarily in the right places for our purposes. So they're good for telling us where populations are, what the air quality is there, not necessarily completely relevant to what's happening in vineyard locations. So we use, as a matter of course, in our research smoke exposures, we use these particle counters from TSI. We measure all different particle sizes. We look at the density of the smoke that we're applying. We're also collecting samples of the smoke so we can measure what's in the smoke. So our system is pretty well characterized. What we wanted to see is, can we use some version of this and put them in commercial vineyards and have the ability to know what's happening in vineyard locations, not just what's happening in Sunnyside or Toppenish or someplace that may or may not be very close to the vineyards we're concerned about. And the monitors that we use are pretty expensive. They're between $5,000 and $6,000 a piece. But there are lower cost monitors out there. There are some relatively inexpensive ones, the Blue Sky, Purple Air, things like that. The issue with those sometimes is the quality of the sensor. And then again, we don't have control necessarily over where they end up being placed. What we wanted to do was find reasonably priced sensors with good quality sensors and be able to put them in commercial vineyards. And that was part of a grant through Wine Commission and then also funded through our SCRI to put these in place. And so the picture on the right is one of the sensors we started with originally, made by Thingy. And we put a bunch of these out. The idea was we would have them in commercial vineyards. We could get real-time information about what's happening for growers and for wineries. And we could use the data to help develop better models for smoke forecasting and risk management. And so this is what one of the stations looks like. This is the one at the Rosa Vineyard that WSU runs near Prosser. Has multiple sensors, PM1, PM2.5, and PM10, so different size dust or smoke particles. Chemical sensors to look at ozone, SO2, nitrite, CO, CO2, a little weather station, and then these were connected to a network that would allow us to look at the data from any place we happen to be. This system is still in place at the Rosa. It still works. It's still in use. We put 8 of these out in commercial vineyards in Washington in 2021. We added 14 more of them in 2022. And at the same time, similar kinds of networks were being established in Northern California and parts of Oregon as well. And the data from these it goes to some of the folks that are on the SCRI grant doing air quality forecasts. There were some issues with these sensors. The early ones were fine. Some of the ones that were being built during COVID, the quality of the sensors is not as good as it was initially. And so we've had a higher failure rate with some of these. So in this past year, we started deploying these sensors. This is a little bit different system, just because we were having issues with the original ones and also issues with just getting them. So we've started to build our own. These ones are being built at Oregon State using a design and some help from Von Walden's group here at WSU. And we started deploying these in Oregon, Washington, and California this past spring and summer. At the end of the season last year, we had 15 of these in place in Washington, plus 7 or 8 of the Thingy sensors that were still working. So we had a total of about 24 sensors out. And again, providing data about what was happening during events, so that we could have an idea when we have an event, what the extent of it, what the density of the smoke was, that sort of thing. So these were out there along with some of the Thingy sensors. The plan is to continue to build more of these and deploy them in more vineyards. We actually took all of the sensors in Washington out of the vineyards at the end of the last season and took them down to OSU in November. They've got some updates, some upgrades to the sensors and to the system that are being installed now. And then we'll collect them from Oregon State and redeploy them into the vineyards they were in this past year, And that'll probably happen in March or April once they've got the updates made so that we have these systems in place for the upcoming growing season. So this will be a continuing thing that we're going to try to expand the number of sensors in the network and also make sure that we keep up to speed as better equipment becomes available. The other goal is to have these all online so that we can access data in real time wherever we happen to be. So that's still in the works. That's still being supported by the SCRI grant. I want to turn now to mitigation in the winery. So this is work that Charity Mausa, who just graduated with her master's, this was work that she's been doing over the last 2 or 3 years, using a combination of ultrafiltration and immobilized enzymes to treat wines that have been exposed to smoke and have negative impacts from that. So the idea is we use instead of doing RO and using a 200 molecular weight cutoff and just creating a permeate, filtering the wine to create a permeate that contains free glycol, free 4-methyl glycol, the cresols, we're using a larger membrane cutoff. So this lets things up to 1,000 molecular weight through. So this filter would allow glycosides plus the free compounds, so free and bound glycol, 4-methyl glycol, et cetera, through the membrane into the permeate. And then once we've got the permeate that contains the glycosides, we can then use immobilized glycosidase enzyme to cleave, to hydrolyze the glycoside, release more glycol, 4-methyl glycol. And then the third step in the process would be to use an adsorbent to remove the glycol, 4-methyl glycol, et cetera. So if you're familiar with using RO for cleaning up wines that have been affected by smoke, you would use a 200 molecular weight cutoff, create a permeate that just has the free compounds, and then run that through a carbon bed to remove the glycol. This is essentially very similar, but we're using a larger pore size on the filter, so we get the glycosides as well. And we have another step in the middle where we're hydrolyzing the glycosides using enzymes. And we, in Charity, I say we, Charity, looked at treating both the permeate and the wine, so looking at just treating the wine with the enzymes versus treating the permeate. The plan over the long term is to get this to be a continuous system where you just connect it to a tank and you just cycle the wine through it until it's cleaned up. But just to be able to characterize the system, initially she was just creating batches of permeate, treating them, and then looking at the outcome. And she, at the very end of her project, had graduated on to doing multi-step where she'd treat the wine to create a permeate, treat the permeate, add that back to the wine, and do multiple steps, but in a batch system so that she could characterize everything. And right now we're using activated carbon to remove the compounds. We're also looking at the potential to use molecularly imprinted polymers that are a little more selective for the things we're trying to remove. So that would be another step we would work towards in this process. So what we've done so far is look at permeate versus wine, and then the batch and activated carbon. These last steps are still remain to be done. I have a new graduate student who just started at the beginning of this month who's going to pick this project up and carry on through getting this to a continuous process, and then eventually looking at using MIPS for removing the glycol, et cetera. Again, what we're trying to do using these enzymes is just cleave the bond between the sugar and the glycol so that we can then remove the glycol with either carbon or MIPS. I don't think we need to spend a lot of time. She was treating both wine and permeate using the enzyme hydrolysis. She looked at a range of different temperatures to find the optimum for the enzyme. We were running free glycol, 4-methyl glycol analysis. We're also doing acid hydrolysis before and after our treatment so we could look at the total pool, the glycoside pool, plus the free compounds. And what she found, or what we looked at, is in black is the free glycol, and then in the burgundy color is free plus bound. So this is what we get from acid hydrolysis. This is what we get from the free analysis. And doing this both with the wine and the permeate to get a sense of just how much of the glycosides we're able to cleave in a single pass, and then how much remains to be treated in the subsequent pass. She looked at temperature dependence from 15 to 45 degrees C. Most enzymes work better at higher temperatures, at least up to a point, and we found that here as well. 35 degrees C, when we're looking at treating wine directly, we'd see a maximum of about 24% hydrolysis in a single pass. With wine, or with permeate, so if you create the permeate and then treat that using the enzymes, we could get up to about 33% hydrolysis. Again, 35 degrees C being the best temperature for doing the hydrolysis. But with permeate in particular, even at 15 degrees C, we're still seeing 25 to 30% hydrolysis. So we are getting the cleaving of the glycosides that we're looking for. So this was all promising work from Charity's project. Again, we were seeing 25 to 33% of the total bound phenols getting hydrolyzed using this enzyme. And she was trying to figure out how much we were getting at each step so that she could start to get an estimate of how many steps it would take to be able to fully break down the glycosides. Now, we're not necessarily going to continue to get this high rate of hydrolysis. So one of the things she was looking at is, does that rate change with additional steps? Does it go up or does it go down? So she graduated at the end of last summer, but she came back in November and December to run some additional trials. In this particular case, she was looking at doing 3 cycles. So if you look at the chart on the right, we have in blue the original wine, and this is free glycol. We're at about 19. And then after the first pass through, she was down to 17 or 18. Second pass, third pass, by the end of the third pass, she's down to about 9. So she went from 19 to about 9 for free glycol in 3 steps. And again, this is just using permeate. So the way she was doing this, she would run the wine through the RO system, create a permeate, incubate that with the enzyme, and then run it through the carbon bed, and then recombine that with what was left of the original wine, the retentate, and then do the same thing again for the second pass and for the third pass. And so you can see, for looking at the free marker compounds, after 3 cycles, we're dropping for glycol from about, let's see if we can get this, from about 19 down to 9, so about 50% reduction. 4-methyl glycol, the same sort of thing. She's going from around 12 to 3.5. And with the 3 cresols, meta, ortho, and para, we're dropping from combined total for the 3 from a little less than 5. By the time we're through 2 passes, it's essentially gone, and there's none at all in the third pass. We looked at the individual cresols. We looked at them individually. I've just combined them here so that the total is more or less on the same scale as the other things. So we're seeing some success in removing free smoke marker compounds using this approach. She also did acid hydrolysis to look at the totals. And here, total glycol in the original wine was about 180, 181 micrograms. After the first pass, it only dropped a little bit, couple of micrograms, but second and third pass, she got down eventually to 135, so about a quarter of the total glycosides were hydrolyzed. Similar results when you look at 4-methyl glycol glycosides, and the meta-orthoparacresol ones, we're seeing 20 to 25% reduction in the glycoside pool. This is consistent with what some other groups have seen. We were using, as our enzyme, we were using just the monoglycosidase from almonds, just a beta glycosidase that primarily focuses on removing glucose and just a single glucose. But as we know, the mix of glycosides that are present in smoke-affected wines includes both monoglycosides, but also diglycosides. And this enzyme doesn't necessarily have very good activity against the diglycosides. And so I think there's a limit to how much we can remove with this particular glycosidase enzyme because it's active primarily against monoglycosides. One of the things that we'll be doing moving forward is looking at other glycosidase enzymes to see if we can get the ability to remove some of the ones that remain here after treatment with the monoglycosidase. So this is still a work in progress, but I think the fact that we are seeing 20 to 25% reduction in the glycosides, as well as a more significant loss in the monoglycoside, says this is a reasonable approach for doing this. What it may take is a cocktail of enzymes that are mobilized, some that work on the monoglycosides, some that work on the diglycosides. So I have a new student who just started, Madison Causey, who's going to continue this project. We're also planning to shift. We'll continue to do acid hydrolysis to get total glycosides, but we're also going to now measure individual glycosides using LC triple quad or LC-Q-TOF-MS, so we can measure the amount of individual glycosides. We started with acid hydrolysis just to give us the total pool, but we want to really pursue this idea that we need enzymes that treat both the monoglycosides, but also the diglycosides, and this will help us understand what specific enzymes are actually affecting each type of individual glycoside. So it's very much a work in progress, but I think there's good progress here. And I think the other step that Madison will be working on is taking this from multi-step to an actual continuous process where the permeate is created, it passes through some immobilized enzyme beds, and then passes through carbon. What comes out of that gets recombined with the original retentate to make a wine that is intact, but minus the glycosides and minus the free guaiacol. And we'll be looking closely at, because we know some glycosides are relatively stable, which glycosides are being removed by which enzymes, and really trying to focus on hydrolyzing the glycosides that contribute to the problem and not maybe worrying as much about some of the glycosides that are stable over time and aren't going to contribute to the problem. So that's why we're shifting from just looking at total glycosides to looking at individual glycosides. A couple other things. We looked at the impact of color. The 1,000 molecular weight cutoff filter will allow anthocyanin glycosides to pass through. So we were concerned that we would see loss of color. And you can see on the left here that we do see in the permeate that we see some color. There's some pink colors indicating there's anthocyanins there. Once you pass it over the carbon bed, those anthocyanins get stripped out by the carbon. So there's the potential to lose some color, and we'll focus on that a little bit in subsequent trials. But if you look at the wines, and one of the things we'll do is look more closely at characterizing the color of the wines before and after. The color is probably a little lighter after 3 cycles, but it's still, I mean, it's still, at least in this picture, it looks like reasonable color. Going forward, we'll characterize this color better so we can actually quantify the extent to which color is being removed by this process. And then if we need to, we'll look at changes to the system that might help us retain color better. But I think at least initially, it doesn't look like it's going to be a significant loss. But we'll keep an eye on that as we develop this further. All right, so I want to shift gears a little bit, talk briefly about the work we've been doing with RNA transcriptomics. This study, as I said at the beginning, was initially funded with seed money from Wine Commission. And then using that money, we're able to include this in our proposal to USDA ARS. And so this project's now being supported by that. Initially, the work was with our lab and with Amit Dingra's lab in Pullman, along with Stephen Ficklin, both in horticulture at the time. Amit's now moved to Texas. So we're working with Stephen Ficklin, and his lab is doing the RNA work. Our lab will be doing the metabolomics part of it. But what I'll show today is original work done with Amit Dingra's lab in 2021. And the idea is we're going to extract RNA from pre-veraison and post-veraison grape samples that we collect during our smoke exposure trials, and we collect them at regular intervals during the smoking so that we can understand during a smoke exposure what genes are being upregulated, what genes are being downregulated, how is the vine responding to the presence of smoke. And so we're looking at RNA taken from samples collected at different points during the exposure and looking both in unsmoked control fruit and in fruit from our smoke exposure trial. So comparing transcriptome from unsmoked fruit to smoked fruit. We have a paper published just the end of last year that shows the first pass with this data from 2021. And we're not going to go into this in great detail, but one of the key things we found in this study is within the first 2 hours, gene expression is changing from the presence of smoke. So the response to the vine is quick. Within 2 hours, we're seeing pathways being turned on, defense mechanisms, things related to vine stress get turned on very quickly. This particular case, we're looking at a glycosyl transferase. So this would be one of the enzymes that's making glycosides of smoke-related compounds, or at least has the ability to do that. And we see a little response early in the first 2 hours, but by the time we get to 6 hours, from 6 hours to 12 hours, we see a rapid increase in the expression of this gene, both in pre-veraison samples in the darker lines, solid lines, and in post-veraison samples, which are the solid lines, but darker color or lighter color. We see a little bit of the response in the unsmoked fruit, but much bigger response in the fruit that's affected by smoke. We see a decline after 24 hours, and then it starts to increase again. And we go as far as 36 hours in the first year. That's the end of the smoke exposure trial. That's where we stopped. In subsequent years, 23, 24, we've continued to collect samples after the exposure ends so we can start to understand when the smoke clears, how does the vine unwind all these systems? How does it return back to normal after the smoke is cleared? How long do these enzymes stay at elevated levels before they start to decline? But so this is one of the glycosyl transferases. We see a rapid increase in the first 12 hours. And this is consistent with what Kerry Wilkinson reported in that study looking at kaolin. In the first 24 hours, most of the fruit has been glycosylated. Here we see in the first 12 hours, we've got a peak of expression of this part of the genome. So we're definitely seeing a vine response that includes glycosylation, and it's happening on the same sort of timeline as we would expect based on Kerry's results from her study in 2019. Now we're not advancing. There we go. One of the other genes we looked at were glutathione transferases. And this is another plant defense mechanism, is to use glutathione. You put a glutathione on xenobiotics as a way of signaling this is a problem, it needs to be dealt with. And we see similarly, we see the glutathione transferase genes, and this is just one of several, being upregulated. Again, you see it start as early as 2 hours, and really at around 6 hours, you start to see this climb to a peak at around 24 hours, and then it maintains fairly high levels with this particular gene out to 36. There are some differences between how the vine responds pre-veraison and post-veraison. So these are all things that we're continuing to look at, how these things get upregulated. And then again, after the smoke clears, how are they turned back off? So a little more basic research, but I think it helps us understand what the vine response is, which will be useful for us in understanding when we need to be most concerned about smoke, how long, for making decisions at the end of, if we have a smoke event during harvest, how long do these systems take to glycosylate to make thiophenols or whatever they're doing? In this particular slide, the top row are 3 different glycosyl transferases. The expression pattern is not necessarily the same from one to the next, but they all respond to the presence of smoke. The 3 across the bottom are 3 different glutathione transferases. And again, in the smoke-affected fruit, we see a response. The pattern isn't necessarily exactly the same, but you do see a rapid response over the first 6 to 12 hours to 24 hours. You see the vine responding to the presence of the smoke by upregulating these genes associated with 2 of the major plant defense mechanisms. So this work is ongoing. We'll continue to work on this. We have an idea. This is just a cartoon that shows kind of how this is working. You have the volatile phenols in the smoke. They end up in the berry, enter through the cuticle. Inside the cell, these compounds are either glycosylated by the glycosyl transferases, or you form a glutathione adduct using the glutathione transferases. This leads to the glycosides that we are also familiar with. We think this system ultimately leads to the thiophenols that we recognized as part of this issue 2 or 3 years ago. We think this is part of how they're produced. We haven't worked out all the details. Part of this will come from the metabolomic study that's ongoing as part of this. But we think this is part of where the thiophenols are coming from. Again, this project is ongoing. These results are for 2021, but we also have similar data being processed. 2022 is largely extracted and out for analysis. 23 and 24 are being analyzed now. And the metabolomics part of this that looks at how the metabolism is changed, what are the compounds that are changed as a result of the transcriptome upregulating or downregulating different parts of the genome. So this project's ongoing, largely supported by USDA ARS funds. So, all right. So the next thing, and really just kind of wrapping things up, just an update on how funding sources are going. Congressional funding through USDA ARS is ongoing. We're in year 5 of that now. And that continues to be provide support for me for half of my technician salary. It pays for 2 graduate students, provides supplies and support for things. It also pays for part of my postdoc. It pays for a lot of the transcriptomic work. It's a tremendous help to have this support. We also have collaborations now with 3 different teams at ARS, in Corvallis, in Albany, and in Berkeley. So we're seeing some headway with making collaborative studies with ARS as well. So I think there's real benefits there for working together with those teams. We also have the USDA SCRI grant. This was a total of about $7.5 million, split between Oregon State, Washington State, and UC Davis. It funds about 16 or 17 researchers. We're just starting year 4 of 4, and we'll be working on the renewal proposal to get a second go around with SCRI support. Again, this pays for technician salary, this pays for graduate students, and also provides support for a lot of supplies and things that we need to do this kind of work. And of course, we have continued to have great support from Washington Wine Commission, Washington State Grape and Wine Research Program, which has been supporting this project since its inception in 2016. So we've been able to leverage that initial funding to make a lot of progress. So that really focuses on where we're at. We will be looking at doing another USDA proposal here so that we can continue this source of funding. I just want to end with just a recognition of people that are involved in getting this done. If we're going to go out and do smoke exposures for 36 hours at a crack 4 or 5 times every summer, it takes a lot of people. Colleagues that have been at WSU that are part of this, some of the USDA scientists, postdocs, graduate students. I mean, there's a whole list here of all the people that have been involved, as well as recognition of the sources of funding for this. Currently, this is not everybody, but this is the crew. Currently, I have one postdoc, 2 PhD students, 2 MS students, a research technologist, and 2 undergraduate interns. We just graduated 2 MS students in the last 4 or 5 months. Jonah here is an MS student who'll probably finish up later this year. Leighton on the left is my postdoc. Mackenzie is one of the PhD students. Cassidy is currently my research technologist. It's a great team. And I just want to recognize everything that I'm talking about. These are the people that are doing the work. So thank you for your support, and we'll keep at it. So I think we have time for questions, right?
Yes, great. So we do have a couple. The first, Tom, is there a list of locations for the sensors in the vineyard? And is that data available to wineries in real time?
We do have lists. The data, so there has to be some sensitivity about how we share this data. Currently, it's being shared primarily with researchers who are part of the project, the folks who are doing the modeling. There has to be some sensitivity to how we share the data more broadly. We want to get to the point where we can share data that is, I guess, sanitized, anonymized, so you know generally where the sensor is, but not exactly, because we don't necessarily want everyone out there to know what's happening in specific sites. We don't want to damage anyone's business. We don't want to create concerns that aren't real. But the goal is to make this available in a way that helps us understand where risk is, but without, I don't know, without creating alarm or creating problems for specific wineries or specific growers. That's something that I think the West Coast Smoke Exposure Task Force can play a role in helping us understand what's the best way to make this information available so that it's there for people to use, but not likely to cause damage to a specific vineyard or a specific winery. I mean, it's sensitive if you've got smoke in your vineyard. That's reason to be concerned. And so we have to figure out the best way to share this without creating too much concern.
And I might add, we also have to figure out what that data means. And
Yes.
so we're not to, this is still research. And so we're not to a point yet that when Tom gets a reading of 10, he knows, oh, those grapes are going to have a problem. We don't know that yet. So
Yeah.
he's working on that. Okay.
Yeah. And smoke is not necessarily smoke. The smoke density is one thing, but the composition of the smoke is another thing. And you can have smoke that's fairly dense that doesn't cause a problem, and smoke that's fairly dense that does. And we're still working out how exactly that works. What are the factors that matter?
Okay, next question. What specific enzyme products were you using, and did you trial different enzymes?
So the enzyme that we used for what I showed you today is just a simple beta glycosidase from Sigma. It's probably the most widely available glycosidase out there. And we started with that one, knowing that it wasn't necessarily going to be able to cleave all of the glycosides. But the first step is really showing that we could use it to cleave any of the glycosides in this kind of system. Now that we've shown we can do that, the next step is to look at other glycosidases that are available and work out a set that helps us cleave the ones that matter. And maybe we won't be as concerned about some of the ones that seem to be stable and don't necessarily cause problems for us. So the first step was really just showing that this approach, this system of using nanofiltration followed by immobilized enzymes would actually work. The next step is to look at other enzymes to give us a better set that will cleave more of the glycosides. So
Yeah.
still a work in progress.
And they are, the ones you used are commercially available.
Yes.
Okay,
Yeah.
that was a question. Okay, another question. Is smoke absorbed just by the grapes or also the leaves?
Smoke is absorbed by both. There's a bit of a debate about what role the leaves play in what ends up in the fruit. So work early in 2010 from Australia showed if you put deuterated or marked glycol on grapevine leaves, you will get deuterated glycosides in the fruit. But they also said that's not the major route. Most of it comes from direct exposure of the berry to the smoke. We did some work over 2022, 23 that showed that the leaves do seem to play a role, a bigger role than they had been given credit for, which is why in the 2024 we went in and looked at putting kaolin on the leaves and the fruit, or just putting it on the fruit. So we could try to see, do we need to cover everything, or is it enough just to cover the clusters? So the short answer is leaves absorb just as well as the fruit does. We're still trying to understand how big a role they play and what ends up in the fruit.
So is there less smoke absorption before veraison than after? Have we really answered that?
We have not really answered that. I would expect there'll be more absorption post-veraison, if for no other reason than the barriers are bigger and the canopy is bigger. What we have shown is pre-veraison smoke can result in smoke-affected fruit. Smoke exposures as early as pea-sized berries can result in fruit that's exposed that has significant impact at harvest time. So the nuances, the details we're still working through, but we know pre-veraison can result in smoke-affected fruit as well as post-veraison does. That's in our studies. But because we do see pre-veraison smoke exposures fairly, I won't say regularly, but often enough that we know it's an issue, we've looked at it and it definitely can be an issue.
Okay, great updates. Would you mind sharing more information about the thiophenol formation pathway? Any work done yet to investigate the formation during winemaking process besides in the grapes?
Well, we will certainly keep everyone updated as we get through the RNA transcriptomics and metabolomics study, which is where we're really trying to understand what's happening in the fruit. The fact that these glutathione transferases get upregulated is consistent with formation of thiophenols in the fruit. This would be a mechanism for getting there. The details are still, I mean, we're going to be looking at in the data from 22 through 24. And we'll have the metabolomics data that shows when the thiophenols are being generated. We know they are generated in the fruit. We've done some other work looking at what happens during fermentation, because it's at least possible that yeast play a role in this as well. The first studies are sort of inconsistent. They don't really show differences by yeast strain for thiophenol formation. Maybe a little, but not enough that we're ready to say, yeah, there's clearly a role that yeast play. But it's, that's ongoing work. That was what Megan, who graduated just in December, was working on that project. As we learn more, we'll certainly put it out in meetings like this or at either WAVE or WAVEx. We'll make sure that information is available.
Okay, here's a couple thiophenol questions.
Okay.
Copper has a well-known ability to react with sulfur-containing compounds in wine. So
Mhm.
have you looked at the impact of copper fining post-fermentation on concentrations of thiophenols in smoke-affected wines, and what impact might this have on sensory?
We've done some very preliminary work looking at whether copper fixes the problem or not. One of the key things in that process is that the thiols readily convert to disulfides, which are not sensitive to copper. So we would have to do any kind of a copper addition for removing thiols would have to be done when those thiols have been reduced from disulfides to thiols. In wine, in most stages of production, there is likely to, in fact, more likely to be disulfides than thiols. So we'd have to address that issue and make sure that we get them converted to thiols first. And it's one of the things that we know we need to look at. We just haven't gotten to really understanding that yet. That's one of the things on our list. We're starting to really look at the role that redox plays or could play in how these compounds play out in wines during aging and during bottling or post-bottling. But it also, redox may play a role in how they're produced. If yeast play a role in this, the redox state during fermentation may be part of the equation too. That study, that's in our new USDA proposal to go down that road. We just haven't gotten started on that yet.
Okay, I think you kind of touched on his other 2 points. Heard you were doing a little yeast stuff. Did you notice any phenotype-genotype relationships between the strains associated with higher thiophenol levels and strains and the lower TP producing strains that could be beneficial in yeast screening or breeding programs?
The first, I mean, I guess what I would say there is we're in early days in that work. The first study, we didn't see big differences, but we also didn't have any redox control during those fermentations. And so we're going to go back and look at that again with a better control over the redox level during the fermentation to see if that plays a role.
Yeah.
Yeah, we're just, in first pass with no redox control, we didn't see big differences. But that doesn't mean with proper redox control, we wouldn't see differences. We're just not there yet.
Okay, and you got the last question about redox control. Okay, how early have you treated with the barrier spray? Was it would earlier than pea size be okay?
Um, we have not done our barrier spray trials because we're running multiple things in any given year. Barrier spray trials have tended to be either right close to veraison or post veraison. It's one of the things we could do is look at doing a barrier spray in one of our first sprays. We haven't done it yet. It's typically been, I mean, we start doing smoke exposures right around the 1st of July, plus or minus a week, depending on berry development. We try to get in there right after bloom, even before we get to pea-sized berries if we can. There was one 2022, I think we got in. We had clusters that had flowers, had pea-sized berries and everything in between on the same vine. And in that year, we still got smoke-affected fruit. We just haven't done barrier sprays that early. That's certainly something we could do. We just haven't done it yet.
Okay. And good to know you're working on this. That was a lot. So any other questions? If not, just a reminder, I did record this and it will be sent to all those who registered, so you'll automatically get it. As well, we will post it on the Wine Commission's website. We have, under the Research and Innovation page, we have a whole section devoted to WAVE, so it'll be on that page as well as in the research library. So with that, I do believe we will be holding another Smoke Summit this June, so keep your lookout for that. And when you have time, not in the heat of the season and harvest, go in and check out the smoke exposure website. As I said, it's a work in progress, but we got all the resources that are current and updated for right now so that if a smoke event happened, growers would have one place to go, and wineries, one place to go to find out, what do I do? So we will be adding to it, as I said, all the research updates and information as they come out. So with that, thank you very much, and
Yeah, thank you.
sign off.